Okay, in our last video, we want to remind you that the topic of this unit is protein function, and the theme is that the structure of proteins is what determines their function in the biological organism. We've already talked about globular water-soluble proteins like myoglobin and hemoglobin. We've talked about structural and fibrous proteins like collagen. And lastly, we want to talk about membrane proteins, which is really a third class of tertiary structure. Again, very different than the previous two. Let's just review quickly what we mean when we talk about membranes. Membranes are bilayers of amphipathic molecules. Remember, amphipathic means molecules that have a very strong charge on one end, very strong polarity, and non-polarity on the other end. So what we end up with is nonpolar tails coming together due to the hydrophobic effect, and the polar heads interacting with water on either side of this sheet that goes on and maybe surrounds a cell. So what if we want to have a protein embedded in the membrane? Instead of being water soluble, it's going to be stuck in the membrane. That's a very useful thing, and we're going to see it a lot throughout this class. The answer is that we have to have the protein have a different coating on the outside. Remember, globular water-soluble proteins had nonpolar on the inside and polar on the outside. And that's somewhat not true in the case of membrane proteins. When we have a membrane protein like the one we have shown here in red, the outside, at least what is facing the membranes, is going to be nonpolar. Remember, those are alpha helices, and they have side chains radiating out from them. So in order for this to be comfortable or stable inside a membrane, we have to have nonpolar things like phenylalanines and methionines and valines poking out and coating that protein so it's comfortable to be embedded in this nonpolar portion of the membrane. That said, you're still going to have polar portions facing outward. Those are going to be the ones that are kind of facing the water on either side of the membrane. Now, in certain cases, for certain membrane proteins, you actually have polar on the inside. And that's the case for this particular type of protein. It's got polar on the inside, and the reason for that is because its function, this particular protein is a function to allow potassium ions with their full charge to pass through the membrane from one side to the other. Remember, we said it's almost impossible for potassium or any charged molecule to go through the membrane itself because it would have to be dragged through this nonpolar area, which polar things hate to do. But we have a pore going down through this protein, and that makes this a protein channel, and it allows the potassium ion to go through the membrane because the interior of the protein is polar. In fact, it's somewhat negatively charged. We can see that a little bit if we take this protein and kind of rotate it toward us. So if we were looking down on the membrane, let's say this was the top of the membrane here, and we were looking down on it, what you can see is there's a beautiful pore right in the middle of it, and that's where the potassium ion would go down through the membrane. And so that's just a really nice example of a membrane protein, and it serves a particular purpose. It's called a potassium channel because it allows the potassium positive charge to channel through the membrane and get to the other side. And these play extremely important roles in biology because a lot of charged molecules, whether they be drugs or ions or things like that, nutrients have to get through the membrane, and they do so typically through these types of proteins. So the type of protein we have here has a particular structure, nonpolar on the outside and polar on the inside, that meets its function, and it's very different than what we saw with the other two classes. It's just an example, but it's a nice one to get to a lot of different proteins that have this similar function.